Okay. Yeah, great. Looks good. So everybody, when you're presenting, stand in front of the computer so your face will show up and it will nicely show in the YouTube in the future. So just let everybody know we're recorded by Zoom and hopefully you will find your own presentation online via YouTube. So we are finally getting into our last day and really congratulate everybody for your effort and for your contributions. Also, we want to thank all the members, the mentors, the lecturers, our admin. So there's and also our big human program director and everybody to make this happen. And also thank for the parents for driving your students every day, come and forth, back and forth to our site. So this is actually our fifth year holding the human site at the Vision as one of the major of the 8 sites here. So in addition to a lot of focuses on cancer, our sites are looking at the unique organ, which is the eye, and also part of the visual functions are also studied in the brain. So here are some pictures I want to share. That's mainly focusing on our first week when the students are really doing amazing jobs, followed up by Kate Davoli's instructions of trying their first bio experiments, trying to learn how to use pipette, how what the eye looks like in real. And also I have some showcase pictures of our roundtable time. Hopefully everybody enjoyed your summer. And also here are the people we really need to thank. Um, so there is first Kate Davoli, who has been with us for over 2 years, and we now finally build up a curriculum for the first week wet lab training, and we found it really helpful for bringing all the students together and let you make friends with each other, let you have a kind of a more formal training of learning how to use pipette and the general anatomy, how to use lab notebook and reports and so on. And also I want to thank all the speakers for the lectures. I give really a great introduction about vision research, including the development of the eye, some infectious disease in the eye, the glaucoma, what glaucoma is, what optic nerves are doing, and also how visual perception is processed in the brain. So, and also a lot of applaud for the mentors who are really devoting a lot of the time in the summer. Really, it's probably you don't know, but most of the mentors here have, many of them are having their first experiences in training and high school students. It's a lot of devotion of time and patience and the resources for opening for high schoolers and with a lot of concern of safety and tailor the training to the level that you need. And also, we want to thank Ioneer Foundation for the funding. So the research is currently funded by Human Foundation as well as the Ioneer Foundations with the strong support of our chair, Dr. Sahel, and our vice chair, Dr. John Ash. And we want to thank David Boone and Stephen Jones for the big umbrella of the Human Cancer Center summer program. And by the end of final day, you will hear in the afternoon for a formal ceremony of the closing. And you will hear like this is a lifelong relationship with the program, and you will be one of the human alumni. And we are providing continuous support if you want to come back to the labs, you want to continue your research training, or if you need reference letter for your college application, or you want advices for your career, you are always welcome to reach out either to the big program or to us. And we also want to thank our admins, Beth, Bake, and and Don, who really provide a lot of back of the scenes when you are receiving your fellowship and your your ID to get inside the building, and many of those things you may not notice. And we want to thank our photographer, Kelsey, for professional photography today, and the Human Foundation. And most importantly, we thank you for your taking advantage of this program. And hopefully that will help you learn more about what vision research is, and hopefully find this is an interesting field in the future. Either you are want to pursuing academic research, or many of you are pre-med. So hopefully that will help you in the future. So I will stop here, and our format. Okay, one more thing. Keep in touch. So many of you are applying either this fall or next fall, and we have already one graduated and going to college. Please let us know, and let the big program know. There will be a follow-up survey, as what John said, and give us feedback. What do you think about the program? What do you think we can improve for the program? And also let us know where you are going, and it will be really a rewarding feeling for us, and allow us to celebrate with you when you really find your college. So just let us know. And also congratulations, and hopefully you enjoy the rest of the summer. And I will stop here. And so the format for today is we're gonna have everybody have about 15 minutes, and every mentor will start introducing your student for about 1 minute or 2 minutes, and then every student have about 10 minutes or 11 minutes of a presentation, followed by about 2 minutes of question and answer session. And we're gonna break when 4 of them finish, and have a 15 minutes break time when we can announce some awards. And then the best presentation, best research award will be voted at the end of when everybody finish your sessions. And then I will be announcing that, and followed by lunch. So the shuttle will come at 12:40 today to our vision building to move all of you to the assembly building. So that's the plan. And I also want to share, this is our first time to have a full house in the session. I really enjoy it. So this is the final day. So all the presentations are in this folder. I'm gonna open the first one. It will be Sarah. Yeah. Hey, John, please come by here.
This takes an incredible amount of work to put together and to pull off. And you know, Yunyun actually gets no salary for this. It doesn't account for anything for tenure or anything like that. It's just done out of passion for training and teaching, and for what you guys are doing. And I really think, you know, she's pulled it off, like 3rd year now.
Fifth
year now, okay. And the 3rd year for me. But she's really done an incredible job with this, and it keeps getting better and better every year. And so I just want to acknowledge Yunyun and just say thank you for that.
And all the mentors, I want to keep saying it's really like a voluntary to every mentor to raise their hand and say, I want to participate this summer. But it's really a lot of work for every mentor to think about your projects, and to pair you with senior lab members. So really, it's like I want to share all the credit to everybody. Yeah.
He's very modest.
Yeah. So the first speaker is Sophia. Mentors here. What? Mentors here. Yeah.
So I
Yes.
added a new one that says final at the end.
Okay. So you want a new?
Yeah, so I don't think it's this one.
Okay. Let me brought that up. So I need to refresh this one probably. Or you can use this
Yeah,
one.
this final. The top one.
This one?
Yeah.
Okay. So where is the slideshow? Here. Slideshow from beginning. There we go. Yeah. Good morning, everybody. Sophia spent a few weeks with us in the lab. She's going to explain a little bit of the work that she was involved in. It was a pleasure to have her joining us, and I would like to say thank you for your patience to be with us. We just established the lab a few months back, so we were trying to find a nice project that you could learn something interesting and useful for your future. So, Sophia.
Hey, well, good morning, everyone. It's really great to be with all of you today. My name's Sophia de la Cruz. I'm a rising senior at Hawkins School. And today I'm going to talk a little bit about my project, optimizing the immunostaining protocol to visualize optic nerve tissue. Okay, so a little bit about the background of this project. We were working with neurofibromatosis 1. This is a genetic disorder that causes patients to get tumors on their nerves. And 1 in 5 of these patients get optic pathway gliomas, or OPG. So this is a tumor in the optic nerve. And these patients are usually children under the age of 12. Unfortunately, patients with NF1 OPGs often also experience loss of vision, and chemotherapy can help shrink the size of the tumors, but it doesn't restore the vision of these patients. So in this project, we were really looking to see how we can visualize the glioma in the optic nerve so we can kind of help these patients with that. And looking at this diagram at the bottom, the red arrow just shows right behind the eyeball, that's exactly in the optic pathway where the glioma would be situated. So a little bit about our aims for this project. First of all, we wanted to find a way to visualize NF1 OPGs, and I say without the need for sectioning because there are ultimately 2 main ways that we visualize. The first way is through sectioning, so we do like very thin slices of the optic nerve, and it becomes like a parts of a whole. So we get a bunch of really thin slices and we're able to visualize them all together afterwards. But the other way that we were really trying to focus on with this project was a clearing method. So what this means is that we take the optic nerve and we turn it transparent, so then we can stain and image it and see it all from the inside and get this. There we go. Okay, sorry about that. And get a holistic view of the interaction between the tumor and the tissue. So in that image right there, in the red oval, you can barely see it because we cleared the optic nerve, but that is an optic nerve of a mouse, and you can see the 2 eyes that are still attached to it. So it was really our goal to get this holistic view of the interactions with the axons that are traveling through the optic nerve and how those are interacting with the cells of the glioma. Finally, we were trying to optimize the methods for clearing the optic nerve. So this was really the main part of this project, especially in the immunostaining part of clearing the optic nerve. We were trying to see how we can best carry that out so then we can help visualize what's going on and eventually help the patients. And here at the bottom is a nice visual of what the clearing would look like for tissue. Here is some more about sectioning, since this is something that took up a lot of my time while I was here this summer. So up there is a short timeline. I mostly worked with those last 3 parts, the sucrose, sectioning, and staining and imaging. And so on the left, you see a whole eye of a mouse. And I zoomed in right there on the retina that's at the bottom. It seems a bit damaged, probably because of a problem during the fixation or sucrose process. But this is something that I use to practice sectioning using a cryostat. That's a machine that gets the tissue really cold, so you're able to get really thin sections and have the parts of a whole, as I was mentioning earlier. And then these other images here, and I've outlined them for you as well. these are a whole optic nerve of a mouse. The first time that I tried sectioning a mouse optic nerve, we saw absolutely no tissue. My mentor, Dr. DeLima, looked at them and she gave me the face because there was no tissue. There was none. But the second time it was better, and we see this beautiful full optic nerve of a mouse. And so I was able to get that practice of seeing these full, and then if you see a glioma inside of it, then you are able to sort of visualize it through the sections. Now talking more about our methods. So for our animals, we used an NF1-OPG mouse model. And in terms of our tissue processing, we did a whole nerve clearing method. So we used a cubic R1 delipidation solution, and this is what made the nerve actually optically transparent. And in terms of the sectioning, we did serial sections of the optic nerve with the cryostat, as I was mentioning earlier, so just chopping it in thin slices. And in terms of our results, for the clearing method, we did a 3-dimensional analysis of the optic nerve. So using a light sheet microscope, and that top image right there, that is what it would look like once you image it after staining. So at the very top, that represents where the eyes would be attached, and then the long part is the actual optic nerve as it attaches to the bottom of that optic chiasm. So just to orient you a little bit there. And that's how you're able to see a whole 3-dimensional look of the optic nerve through this clearing method. And that's why it's really useful to us. We can see it in the 3D. And then in terms of our sectioning, it's a 2-dimensional analysis, but a 3-dimensional reconstruction. So using confocal microscopy of multiple sections that I took with the cryostat, and then putting them together in a Z-stack to have a full 3-dimensional look. Now this takes a lot more time, a lot more effort. It can often be maybe not as accurate if you're missing a section as the clearing method. So that's why we're really trying to focus on that, and in this project, optimize those methods. Okay, so moving into the immunohistochemistry, this has to do with the staining and imaging process of the optic nerve protocol. And so our first trial just had a pretty standard incubation in primary antibodies, and then an incubation into our secondary antibodies plus TOPRO. And here are 3 channels. At the very top, we see CC1. This is our red channel, and this is the cell bodies for our oligodendrocytes. So we can see that this was stained. Like if I zoom in there, sorry, it's a bit blurry because I just zoomed into the picture, but you can see that there are the cell bodies that are being seen. Oh, sorry. And then here is a video of going through the CC1 of the optic nerve longitudinally, but the other 2, let me see if I can go back. There we go. Okay. For our OLIG2 and our TOPRO, those are both nuclear. Those are both our nuclear markers. And for OLIG2, it's the nuclear markers for our oligodendrocytes. And for TOPRO, it's just for any cell found in the optic nerve. And we really don't see that those were being stained. So that was something that we wanted to optimize in our second trial. All right, and then our after immunostaining process had to do with just some post-fixation or dehydration. And is it going to play the video again? No, it's not. Well, it's okay. All right, so this is our second trial. It's already playing the videos, but that's fine. So we decided to use a switch buffer system for this, and this basically just helped penetrate through the membrane of the cell and through the nuclear membrane and get the antibodies all the way into the tissue to just maximize permeability. So if I play these again. Okay, so we can see that it is a lot more densely expressed in the TOPRO, and we are definitely getting some positive for staining in our TOPRO and our CC1. It's going to play our OLIG2 here. There we go. Okay, and then in terms of our OLIG2, we don't see it as strongly. This is also probably likely due to an imaging issue, and the reason we see the TOPRO so strongly is most likely because it's the nuclear marker for any cell. So that's why we're seeing a lot more of it. But it seems that our second trial did help improve with that. So let's see. There we go. So in our conclusion, this summer I worked to visualize through cryosectioning or through optimizing a clearing method. And the switch buffer system really increased the permeability of the TOPRO nuclear stain. And we kind of stayed with that permeability with the CC1. Our OLIG2 is not as clearly seen, but this is due to imaging most likely. And this is something that we do want to work on in the future. Unfortunately, if I had more time in the lab, I would be able to spend more time getting better images, better videos to show you. But we did this, it seems that all around we did get a lot of improvement with visualizing that. And some more of our future directions is testing other glial markers instead of just our oligodendrocytes, which are the type of glial cells that we were using as our markers, and to optimize our protocol as needed, whether that is in the clearing process or in the immunohistochemistry process. Okay, and my final acknowledgments, a really special thank you to the site head, Dr. Chen, my PI and mentor, Dr. DeLima, and my assistant mentor, Rusi, the equipment trainer, and who helped us at the beginning, Kate. Thank you so much. And a final big thank you to the Hillman Academy, Stephen Jones, Dr. David Boone. This is an incredible opportunity this summer, so I want to thank everyone.
Yeah.
So that was a really great presentation. Could you go back to your previous slide?
Of course.
So you talk about like testing other glial markers. Do you have any like idea of what glial markers you would also want to test?
Okay, so, well, Dr. DeLima, what are the other ones that are in the future?
So talk about the glial cells,
Mm-hmm.
exercise, also things microglial cells, as in the tumor environment. So they're in the nerve, they are part of the nerve, they're part of the cells from the convolutional nerve. And some of the cells are making the myelin, which is a sheath that protects the nerves and also helps with the conduction of the potential down the nerve. But these cells aren't present. But when the tumor happens, we have proliferation and more of those cells in a kind of immature state and also in a mature state. So we could stain for astrocytes, oligodendrocytes in different stages of differentiation. What's called reference cells, mature cells, nutritional, and microglia.
Next one is Sarah Beth.
So Sarah gets 2 introductions. Of mentors. I first want to say, I've mentors have been around long enough to have mentored many high school students and research projects and such for a number of years. Couple of them are now professors at pretty well-known universities. So these kinds of research experience and programs are great stepping stones for people who are really motivated and more medical and technical and research oriented careers. So it's not just having a great summer, but it's actually developing some skills in critical thinking and learning that I think sets some career trajectories. So really proud to have that. Having said that, Sarah was an amazing student in the lab. Very first time we were meeting, we were sitting around discussing the project, and she asked questions that even third-year grad students might not come up with, like really in-depth critical thinking about what we were discussing. This is really an amazing skill to have at such an early point in the career that I see a very bright future, I think, coming. So really, no pressure. But I see really some amazing things coming up. And I'm going to let Connie, Dr. Podolinsky, finish the introduction because we co-mentored Sarah and Connie did most of the heavy lifting in that program. So I'm going to let her finish.
Thank you, Sarah. I want to say she was amazing. It's not just an amazing person, she's super smart. As Dr. Ash said, she was doing super smart questions and she did an amazing work. She did a ton of work in the lab. She learned how to manage some new techniques that even for trained people, it's not easy sometimes. She did an amazing work and I'm sure she can manage whatever she picks to do in the field. She will be amazing. Thank you so much.
So this is my presentation on protecting the eye, how LIF increases key proteins in the retina. So the lab I worked in focuses on retinal degeneration, and as you can see in this figure, the retina is the thin light sensing tissue in the back of the eye, and it is comprised of different neurons and glial cells. The cells responsible for detecting light and transmitting those signals to the brain are the photoreceptors, and the photoreceptors are susceptible to stress due to different mutations or trauma or environmental factors. And when that happens, it significantly impairs a person's vision. What we know from previous research is that LIF, which is a cytokine, as you can see in the crystal image of it, is neuroprotective against retinal degeneration. It communicates between the immune cells in the eye and helps protect and improve future resiliency to cell stress. And it works through the JAK and STAT3 activation, and once STAT3 passes through the nuclear membrane, it upregulates neuroprotective genes. We also know that LIF activates interferon regulatory factors, including IRF9 and IRF1. IRF1 leads to the transcriptional induction of specific genes related to antiviral effects, immunomodulation, and anti-proliferative effects. This is related with immunoproteasome and antigen presentation, which includes a bunch of different PSMBs. In previous research, we see that the gene expression levels for IRFs increase in LIF female and male mice models. When they are injected with it, you can see that there are different sex-based differences in the IRF expression, and this is the same thing in proteasome, immunoproteasome gene expression, where there is a huge increase in a lot of the units related to the 20S core. So what is the immunoproteasome? The immunoproteasome is a protein complex that is basically a big recycling machine for the cell, and it works by breaking down proteins that are misfolded or mutated and into amino acids for the cell. And the difference between an immunoproteasome and a regular proteasome are these three subunits in purple. Basically become the ones that are shown in green. And it is formed by a bunch of different subunits, including the 11S, the 20S core, and the 19S. My research question, one second. We understand how this works, and it's through the interferon gamma signaling pathway, but we don't really know why this works. And so part of my research question is, can LIF regulate the protein expression of the INF pathway? And my approach was to use different Western blots to measure the protein expression of IRF1, IRF9, and the PSMB8 and PSMB5 in retinal samples from LIF and PBS injected mice. So our methods included taking the samples after 48 hours and freezing them in liquid nitrogen before we use them for our Western blot analysis. For those of you who have never seen a Western blot before, it's mostly comprised of these 4 steps, including the protein extraction and lysis, where we took the retinal tissue, we put it into a RIPA lysis buffer in the protease inhibitor cocktail, and we sonicated it, and so that it was homogenized. We sonicated before homogenization. We then centrifuged the samples, recovered the supernatant before doing the protein lysis and protein quantification using the BCA. We then loaded the proteins into a gel and used electrophoresis to separate the proteins based on their electric charge and their size. We then transferred this membrane gel onto a filter paper using nitrocellulose, and I have this right here if you want to pass it around. We did the red Ponsard staining, which is just to see if the proteins are present or not. before washing that off. We then used the probing with antibodies to detect the target protein, and we used chemi-imaging to detect the bands and visualize everything according to their molecular weight, as you can see in the ladder, which was used as a reference. We also use an automated version of this process known as the simple Western Jess, and it's much faster. It only takes 3 hours versus 2 days. And after taking out our samples and incubating them, we pipette an entire plate like this. As you can see, there's a very small wells. This is mostly the work that I did, all the pipetting. And we centrifuged the samples before putting them into the chemi-imaging machine, which did the chemiluminescence detection and the visualization for us. Some disadvantages to this process were that there are limited antibodies validated for this technique. So it made it harder for us to test certain IRFs and PSMB units, but we were able to use the Western blot to detect signals. It's also highly costly due to the supplies, specifically these cartridges that are glass that take the samples in the machine. So this is the first set of results. These are the IRF antibodies that we validated for Jess. We tested all these different concentration levels to see if we could get the correct bands. And as you can see, we detected a clear signal for IRF9 and IRF1 that shows that they were present in the samples treated with LIF. We also did the same validation step for the proteasome antibodies, and the dilutions for the antibodies that worked for PSMB5 are 1 in 200, for 11S, 1 in 500, and the 20S, 1 in 2000. It's basically a big guess and check system where we test the dilutions to see what works. And we did the same thing for the PSMB units. As you can see, we received no signal for PSMB 8, 9, or 10 in the regular Jess, which is why we moved to, in the Jess system, which is why we moved to the regular Western blot. And we did detect the signal for PSMB 8. So, did the IRFs increase? Yes, they did increase with the LIF treatment. As you can see in both male and female models, there was a significant increase in the IRFs compared to the PBS control, and also you can see some differences in the male and female models. These were done using the triplicates, so there were three R1, R2, and R3 for each PBS and LIF sample. However, the 20S core and PSMB5 did not show an increase with the LIF treatment, and there was no statistically significant difference between the ones treated with PBS or LIF. PSMB8 did increase with the LIF treatment for females, but not in males, so this suggests that there is a sex-dependent difference. And we were curious to find out why, but we don't know. In summary, we know that LIF increases the expression of the transcription factors IRF1 and IRF9. It also induces the expression of the immunoproteasome subunit PSMB8. While the IRF is increased in both females and males, the PSMB8 only increased in females, and it might reflect a possible difference in the protective responses. Our results from PSMB5 and the 20S do not reflect the predictions from the bulk RNA-seq that I showed in the very beginning. And we were wondering why this happened, and we thought it might just not, because our samples were only done with 48 hours, it might not be enough time to see the change in the proteasome assembly, or there might be competition between the PSMB units, so it might not undergo the full change. For our future directions, we need to test the activity of the immunoproteasomes and continue testing other antibodies to understand the mechanisms behind this pathway. We also want to understand the role of the IRFs and the immunoproteasome in a retinal degeneration model, not in the healthy model that we used. And addressing these topics will help to explain how LIF has the potential to be used eventually in gene therapy patients with neurodegenerative retinal disorders like glaucoma, retinitis pigmentosa, and age-related macular degeneration, which is what like John Ash is studying right now, and a lot of the labs we're working on this summer. I would really like to acknowledge my amazing mentors this summer, Dr. John Ash and Doctors John Ash and Constanza Potolinsky for helping me so much and always being there if I needed anything or was confused about something. And to Dr. Chen and to Kate DeVoley, thank you so much for helping with the training at the beginning of the summer. I don't think I would have been able to do any of the pipetting that I ended up doing without you. Thank you.
So did you do both methods?
Yeah, we use both
Which
methods.
one do you think gives the best information?
Which one gives the best information? I think that it would be amazing if the Jess gave the best information because it's highly sensitive, it's really fast, and it works really well. However, I think neither of them are like perfect systems. I think for the Western blot, if you like, it's very easy to mess up or it's very easy to do something wrong. And I think I think they just both need to be improved, and I think the Jess eventually will be better than the regular Western. I would say the Western is a lot more fun though. I really liked the electrophoresis steps and seeing everything change colors and loading the pipettes, and it's more labor intensive, but that's what I really liked about my project was how like hands-on it was. Which
Get bubbles?
I sometimes did. I really tried to avoid them.
Nightmare of every Western blotter.
Yeah.
You hate mustard. I know.
Oh.
Many people dislike doing mustard, but that's exactly. Start making small errors that accumulate and then you find out how it's fun to do.
That's why it's really helpful to have a second person in my lab. Like I was never doing a regular Western blot without Connie, and she always was there in case I did mess up. So that was really important versus like I was almost able to do all the steps for the Jess by myself, which was really nice to have the confidence to know where everything was. So, yeah. It was really hard though.
Yeah.
And everything's clear. So you might like you have to remember you have to write it down as you're doing it.
So are we keeping that as a souvenir?
I don't know. Depends if it can be used or not. I think it's just empty.
But there are limitations.
Yeah, there were plenty of limitations to this study. So there were no positive controls for samples treated with the interferon, and we need to find antibodies to check the PSMB9 and PSMB10 because for both the Western blot and the Jess, we didn't see any signal. And the regulatory subunits should be checked because all of the subunits are necessary for a functional immunoproteasome, not just the ones we found results for. And all the molecules that were checked in this work, as I said before, were from 48-hour samples. We didn't do anything longer than that because we know that the protective response was observed at this time. But that doesn't mean that the immunoproteasome is fully activated and together. Thank you. So it's a great job, like everybody is keeping on time. Our next speaker is Alex Small, and the mentor is Dr. Ethan Rossi. So.
All right, well, it's a pleasure to introduce Alex this morning. So Alex is a recent graduate of the Hillel Academy at Pittsburgh, and he joined our team to work with one of the senior research scientists on our team, Robert Dram, who's also here. Robert's been investigating the inflammatory response that follows and accompanies the onset of age-related macular degeneration. And we set Alex on the task of trying to figure out what we're seeing in these patients as we're sort of, for the first time, with a new technology, we have able to visualize, structures in the eye that are not visible in the clinic. It's really been a pleasure to mentor Alex. He's got a very inquisitive mind. He asks really great questions. He's also got a really great sense of humor, and I think he's really integrated well with the team. Like the whole team came out to support him as well. My whole lab was here to support Alex. I think he really integrated quickly with our close-knit group, and we really enjoyed having him on the team this summer. We're really gonna miss him and look forward to hearing his presentation.
Thank you. Apologies for the paper. We're experiencing a little bit of technical difficulties. Good morning, everybody. Thank you for attending. I'm excited to share what the team and I have been working on. We're studying immune cells in patients with AMD. But what is AMD? To start off, AMD stands for age-related macular degeneration and is the leading cause of irreversible vision loss in older adults. AMD can be split up into 3 stages: early, intermediate, and advanced. Here you can see the life-changing impacts that AMD causes. Central vision is blurred in early stages, partially obscured in intermediate stages, and almost completely gone in late stages. But what does an eye affected with AMD look like? Although manifestations can vary from patient to patient, each stage of AMD typically looks different. Here, we have colored fundus images of the retina. From left to right, we have an example of a healthy retina, one with early or intermediate AMD and 2, each afflicted with a different form of late-stage AMD. The main sign of early and intermediate AMD is the accumulation of drusen, as shown by the white arrows. These drusen are yellowish deposits of lipids, proteins, and cellular debris. These drusen are capable of displacing photoreceptors and causing central vision loss, as seen earlier. As for the 2 late stages, geographic atrophy is when the drusen enlarge and collapse, causing the destruction of the macula and loss of central vision. And neovascular AMD is the formation of leaking blood vessels. We'll primarily be focused on early and intermediate AMD, but it's important to be aware of the later stages as well. This is what we can see clinically. Here's the retina and a side view of the drusen. It's great that we can see it, but what's happening at a cellular level? Well, we know there's an immune response near the drusen, but that leads us to another question. When does this occur? When do immune cells activate? Looking to the literature, macrophages have been observed around large drusen and in areas of atrophy. Those green dots are immune cells, and as you can see, they are especially prevalent in images C and D. We want to better understand when these cells accumulate during the transition from early to later stages of AMD. There are new therapeutic treatments that focus on the immune response in AMD, and we may want to track their progress. You may think that the images above would suffice, and we'd be able to use the same method to see their cells and track their progress. However, these are images from histology, and we cannot obtain this level of resolution in the living eye. Thankfully, we are able to overcome this issue and image patients at a cellular level using a technology called adaptive optics. But what is AO? It's a system that detects aberrated wavefronts from the eye and corrects them using an adaptive mirror and a closed loop. Here you can see a plane of wave of light entering the eye and being reflected. And when exiting, it becomes aberrated by the cornea and the lens. Next, the wavefront is sensed by the deformable mirror and converted into a planar wave. By making these wavefronts planar, our imaging gains more resolution, contrast, and light, massively increasing our imaging capabilities, as seen here. This video was taken by pairing our AO with a scanning light ophthalmoscope. Our AOSLO has several modalities that allow us to see different structures. This is an example of an AOSLO image of the central macula of a healthy young person. We can image retinal pigmented epithelial cells using autofluorescent light. Simultaneously, we can see the spots here, each one a cone photoreceptor. Using the last modality, we can see these dynamic cells that we hypothesize are microglia. Here, we applied these three modalities to observe drusen at different stages. Note the disruption of the cones and the RPE layer as the drusen enlarge. If we use another feature of the AOSLO, the ability to shift focal points and hone in on different layers of the retina, we can see even more structures. For example, putative immune cells. Now that we've seen these, how do we further prove that these are indeed immune cells? And does this mean that there are signs of immune cell activity around smaller drusen at earlier stages of disease? To answer this, we reflect on images that had UPOs, or unidentified phase objects. Our goal was to determine the identity of these UPOs. So we manually measured 137 unidentified phase objects across 4 patients so far. These objects included 72 circular, 59 elliptical, and 6 irregularly shaped. But those measurements were useless unless we had something to compare it to. So we dove into literature, including histological reports, clinical studies, and trials. At first, we thought it may have been components of drusen, such as vitronectin or complement factor H. But these were all the wrong size, so they couldn't be so. So, possibly astrocytes or Mueller glia. But we concluded that these UPOs could not be any of these structures either, as both astrocytes and Mueller glia are the wrong shape. Our current hypothesis is that some of these UPOs are microglia, the resident immune cells of the eye. This is primarily because of their size, morphology, and location. Initially, we believed that the microglial morphology didn't match, as they have processes, the protrusions seen in A and B. However, studies have shown that when activated, microglia enlarge and retract the processes, and when there's sufficient stress, they can even become amoeboid. Sorry. Our claim is that there are more microglia present in early and intermediate stages of AMD than previously believed. If we look carefully, we can see some similarly shaped and sized UPOs in these images. These are the results. Many circular objects were consistent with microglia, and more oblong and elliptical objects were as well. However, not all of these objects were consistent with microglia, as some had different morphologies and sizes. Because we don't know what these are, something we can do in the future is investigate them and determine their impact on AMD progression. Additionally, because we lack treatments that would prevent progression from early to intermediate and intermediate to late stage AMD, we could perform longitudinal studies to help us understand the activities of these cells, such as their movement and phagocytosis, and possibly create new therapeutic treatments. Lastly, we could track the efficacy of these treatments and others targeting the immune response in the eye. I would like to thank everybody. I would like to thank Dr. Rossi, Dr. Dram, all of Rossi Lab, if you could raise your hands.
Wow.
I want to thank Dr. Chen, Kate Davily, Dr. Boone, Stephen Jones, and all of the roundtable seminar speakers. Thank you so much. Any questions? Dr. Ash?
Yes. So have you looked at, considered like some of the histological change for microglia, like tissues and all? Because sometimes they can be migrating like sideways, laterally, and they'll look bigger, but they might also be migrating like up and down the tissue, which they might be smaller. Could you have any kind of classification?
I had not considered that. That's an excellent point. Thank you.
Yeah, they're gonna, I would roll this.
Right. As we saw in the GIF earlier when they were just moving about.
Nice work.
Thank you. All right.
Our next speaker is Rana. Dr. Fuller is the mentor. Oh, it's not. So Dr. Fuller can give a introduction while I'm.
Yeah, so it's my pleasure to introduce Rana, who is an incoming senior at Grand Allegheny High School. So in thinking about what we might have Rana do, I decided to throw on something completely new. And I say new, 'cause it's really a resurrection of some of my old graduate thesis work, genetically characterizing a fungal signaling pathway for infection in a lung infection model. So we had Rana to see if we could target this pharmacologically, potentially as a first step for developing new antifungals. I don't think when Rana signed up for this program, she thought she'd be spending most of her time culturing and looking at fungus on the plate, but she sort of went with the flow. Hopefully she had a good time. And I think the important thing is that she's got results that the lab can build on, which is a good place to be for any sort of scientific project. So it's been a real pleasure to have her, and look forward to her talk and look forward to keeping in touch and seeing what she does next. Thanks.
Hello, so I wanted to talk to you about my summer project, which I did on targeting the protein kinase A pathway as maybe a viable option to finding an effective drug that works for fungal keratitis. So, do I just do this? Yeah. So, what is fungal keratitis? Let's just talk about that first. Fungal keratitis is a corneal infectious disease that affects the outermost part of the eye most of the time, and it affects about 1 to 2 million people yearly. And it is mostly seen in people that engage in activity that affect the cornea and make corneal damage. But also it affects the people in the USA primarily for the usage of contact lenses. And out of the 1 to 2 million cases that happen yearly, about 40% of them need to get a corneal transplant in order to restore their vision. And out of those 2 to 4 million eyes, about 100,000 actually need to be removed. And how this fungus works is that it finds its way inside a damaged epithelium and makes its way down into the stroma and eats away at the collagen and starts growing there. And as our immune system tries to fight the fungus, it only actually kind of just inflames the eye even more, damaging it further. You can see this patient that has been admitted into the clinic that already has an inflamed eye, but after 1 week, you can see that the eye got even more inflamed and it gets pretty bad pretty fast. So why are we doing this research though? Well, I think it's pretty obvious, but I'm just going to tell about it more. I already told you that 40% end up needing corneal transplants, but I think this just goes on to show that the drugs on the market don't work well enough. The drugs such as voriconazole and neomycin are not doing their job well enough for the patients to be recovering. And I did once again say that 40% of the people need corneal transplants, but there are simply not enough corneas to be transplanting to all these patients. And and even if there were, well, the corneal transplant is a highly risky transplant, which means that it could actually just easily get reinfected, or the eye could just reject the transplant. So we need a better drug. And in the Fuller Lab, how we went about finding a way to start and look for this drug is we recognize that we need a drug target. And to find this drug target, we chose pathways. We wanted to choose a pathway that could maybe lead us to find a drug and test them. And luckily, our fungus, Aspergillus fumigatus, actually infects both the lungs and the cornea. And going off from those characteristics, we chose our pathway, which is the protein kinase A pathway. So the protein kinase A pathway, it has a lot of downstream targets, but what we mainly focused on is that the virulence is severely reduced once you delete the PK pathway entirely from the fungus. As you can see from this figure on the bottom, the wild type is growing kind of uncontrollably, but when the PK pathway is knocked out from the fungus, the fungus struggles to grow severely. And if you look at the graph above, which is an experiment that has been done on the lungs of mice, you can see the wild type, which has the normal fungus infected, they end up kind of dying by the 10th day, whereas the PK knockout fungus that has been injected into the mice's lungs, they end up mainly surviving. So we had two key questions that we wanted to answer with our experiment. Does the PK regulate the corneal virulence? And can we find a PK inhibitor and repurpose a drug that is already out there? Well, we kind of focused on the second question as we were doing our experiments, and we chose H89, therefore. H89 is a drug that mainly is a PK inhibitor in mammals, such as humans. But we asked the question, can the mammal PK inhibitor H89 actually inhibit the fungus inhibit the PK in the fungus as well? So we started our study pretty small. We wanted to see the effect of H89, even if it had an effect at all. And we put our fungus Aspergillus in all of these plates, all of these wells, I might say. And then we created H89 dilutions to see at which point the inhibition actually becomes clear and well enough for the fungus to stop thriving. And as you can see over here, at 62.5 micrograms concentrated, H89 actually has a severe effect, a severe inhibition effect on the fungus. So we wanted to observe this physically. We created a Petri dish experiment where we put media mixed with 60 microgram concentration media. And then we spotted a little bit amount of fungus, let it dry, and put it in the incubator. We observed the fungal growth over 24-hour intervals. And as that happened, we took records of the diameter of the fungus to see how well or how bad it grows. These are two different plates that are by the endpoint of the experiment, and you can also, again, physically observe that Aspergillus stopped growing when H89 was used, which means that H89 is doing something to Aspergillus for it to stop growing. And if you can see from this graph, which is the entire experiment over the 24-hour intervals for 3 days, in the first day, we don't really see a big enough inhibition, but by 48 hours, there is a significant increase between the drop of the fungus and its growth. So we kind of proved already that H89 does inhibit the fungus in some way. And we wanted to see, can it work with the market drug that is already being used, voriconazole, in combination to inhibit Aspergillus? So we create this experiment. I know this is going to be a little bit hard to comprehend because there's two variables, but please bear with me. We had our plate, which we put in all wells Aspergillus, the same amount of Aspergillus. And on the left side of the plate, we didn't put anything. But on the right side of the plate, we also put H89. After that, we created voriconazole dilutions and put it into all the rows, as you can see with the values. And after that, we read the plate, and as you can see, the minimum inhibition concentration, which is when the fungus stops growing entirely, is 1 microgram concentration for the side that did not have the H89. But when the H89 comes into effect, the minimum inhibition concentration actually is 0.5 micrograms concentrated, which tells us that H89 does go well in combination with voriconazole and works to inhibit as a sub-inhibitor. So the conclusions that we came to from this study is that H89 does indeed work to inhibit Aspergillus, and H89 can be combined with voriconazole to inhibit the fungus even further. In the future studies, we want to show that PK does regulate the corneal infection by creating eye models one with the pKa knocked out, one without the pKa knocked out, and putting in the fungus to see in which one it grows. And also, we want to prove that HA9 is actually inhibiting the pKa and not some other pathway that we don't know about. And lastly, we want to test that the drug is actually healthy for the eyes and not actually actively killing the eye as it's treating the fungus. I want to thank everyone in this program, especially Dr. Kevin Fuller, who bears with me through the program, Manali Kamal, who helped me through basically everything, Gotham, Josh, who's not here, Abby, and all my friends that made this program more enjoyable, Dr. Chen, for teaching us everything that we did. And thank you. Questions?
So PKA is used very heavily by most cells, all cells.
Mm-hmm.
So are you worried that
It might have an
it
effect.
might be toxic?
We do think we acknowledge the possibility, but H89 is mainly a topical drug for the cornea, which we don't know if it will get into the bloodstream, which is why we want to conduct an experiment on the mice eye to see if it actually does and is healthy for the eye and the entire body in general.
I'm just thinking that it might inhibit the normal cornea.
Yes, that is, yeah. We also consider that we are maybe, possibly, not me, maybe, but the lab is definitely going to conduct an experiment on HA9 and how it affects the eye in combination with PKA and non-PKA to see if it actually does kill any PKAs. We don't even know if there's anything that is happening in the eye with PKA and its pathway, so maybe it just doesn't do anything.
All right, great presentation, Rana. I have a quick question about, can you go back to the slide where there's a picture of the map?
The map?
Yeah.
The first one?
Yeah.
Okay. Is there a way I can go through faster? Nope, okay. It's right about here.
Right. Why do you think that fungal keratitis is more prevalent in some countries as opposed to that?
Great question. As you can see, I listed agricultural workers as a primary case, as a primary patient. These people, they work in farming and they often live in more developing countries such as China, India, so on and so forth, where medical treatment is scarce and maybe it's not easy to come by. Well, these people, they do get corneal damage more, like, you know, they're more likely to get corneal damage because they're out there working on the farm. And when Aspergillus gets in there, they don't notice it because Aspergillus actually only becomes inflammatory after a while as the immune system sends the cells into the cornea. And by the time the inflammation actually starts, the fungus has started growing. And by that point, after, even if they go to the clinic, there is maybe not a way to treat it since the drugs and everything is scarce and they might not be able to pay or provide for them.
So.
Oh, you can say.
I just have one more part. So you're saying like if agricultural activity like decreases, then the cases of fungal keratitis would also decrease?
Well,
if there's less,
might be, but there are other factors like contact lens use, which is if you don't disinfect your contact lenses well enough, you are basically prone to getting any eye disease way, easier. So in USA, which is another place you can see is a little bit more red, pinkish, a lot of contact lenses going on in America. So when disinfectors don't work well enough or disinfection is like just non-existent maybe, then it is way easier to get it even when Aspergillus is not even like that around in America.
Thank you.
cell responsiveness to the drug use, because if.
Immune cell response?
Yeah, if it's targeting the drug, if the drug is targeting fungus and it's targeting immune cells, you can also target immune cells.
We actually want to look for that because we do not yet have any eye experiments, like actual eye experiments that go on. So we do not know what H89 does to the immune cells that come to stop the fungus. If we maybe inhibit the fungus well enough, maybe the immune cells won't even come into the cornea, so it might not even be of concern for us.
You know what happened?
I don't, but maybe Dr. Fuller will know.
Yeah, I mean, some of these compounds are involved in preclinical model information. You know, we suspect that if there's any sort of, some immune system, which is not only, you know, yeah, obviously what we love, but intuitively, but importantly, actually, beneficial with, as the more puzzle work as well as some of the hyper. So, really much the, those are sort of the.
Now we're right at the midway. So while it's kind of a break time, our breakfast is here. So everybody feel free to fetch something to eat. And also, like, I would recommend mentors and your student come up and take photos and hand the certificate for the training. And we also want to announce 2 awards. One is for the lab report award, and one is for best lab note award, right? And
best
there's
question.
another award for best questions award for those who ask really pertinent and smart questions.
I will do that and then you can grab your stuff. So everyone will have a certificate of completion, which explains, you're going to get another one for the overall program, but this explains the things you learned in that original lab week for your records. But there are also 4 actually recipients of these 3 awards. One is for best lab notebook and for her excellent penmanship. And that was not the only criterion. And thorough engagement with optional questions. So all of my lab report or lab activities came with up to 25 just questions to wonder about that were optional. And Jacqueline Tang did an excellent job with all of those. Really good. And for best lab report, you were allowed to choose any of the 5 that we dealt with and then write up a more formal report, something that would be a written version of something like what you're just seeing about their individual products. The best among those was Aubrey Suda. Very professional. I'm extremely pleased. And our 2 best questioners are William Zhang and Sarah Beth Winkhoff. See that already with how engaged they have been with the presentations this morning. Congratulations.
Then
Have some more.
yeah, so then let's hand out.
Yeah,
You
can-
can.
I'll give these to the students, but you might want to like pose for a picture.
Yeah, so when Kate, name your name, please, the mentor and the student come up and we take a picture.
Sophia. And
Kendra, my graduate student who is on her way, she just messaged me that she'll be here in 3 minutes. Kendra did a lot, if not more, mentoring than I did. So I just want to give her a shout out because she's been excellent, but she was a grad student met with excellent super grad student Shipper. But she contributed to that. But that's it. How was my stall? Good.
That's great.
Thank you.
Hi, I'm Aubrey Suter. I go to Bishop Canevin High School and I am a junior. And today my presentation is on understanding real world eye movements. And the real world points to the everyday movements that your eyes create that are very like erratic and instead of in the lab. So why do we move our eyes? Our eyes move all the time. to help us explore our surroundings, know what to do next, follow moving objects, understand what you're reading or what you're looking at, and then also helping to communicate with people. And fun fact, we have more eye movements than our heartbeats. So here is a video, an example of me wearing the glasses and the recording. And then the red dot is the gaze point where I'm looking at in the video. And then as you can see, my eyes are like always moving. So how do we move our eyes? I'm going to talk about three main movements. And the first one is a fixation. A fixation is a brief pause that stabilizes the gaze at one point, and you stop and you hold your eyes there. It helps you gather information. And then fun fact, even if you think you're stopping and looking your eyes at one place, you're still slightly jittering your eyes no matter what. So the next one is a saccade. A saccade is a quick jerky movement. It helps you scan a scene quickly. So just move your eyes around really fast. It can also be reflexive and under your control. Examples of this are reading, searching for a friend in the crowd, or checking a notification. So if you look at this red dot, I'm going to click the keyboard and you will create a saccade if you watched it. That is a saccade. So the next one is, and the last one is a smooth pursuit. A smooth pursuit is a slow, steady movement which helps you track in a moving object. So it could be watching a bird fly from a tree branch to the next tree branch, or watching a car from a stoplight to the next stoplight. If you follow the blue dot with your eyes, you created a smooth pursuit. So the difference between laboratory testing versus real world testing, the eye movement testings in the lab are limited by short recording times. There are narrow ranges of simple tasks. Participants have their head stabilized, so there will not be any deviations of your head. And then the dim rooms where the participants are seated in front of the computer screen. So obviously, if there's a bright light that shines in the middle of the room, you're gonna look at that. But in a real world, it might be different because there could be a loud boom and you look like that way. So that could deviate your head and then your movement will change. In the real world, there's always a specific movement that's happening no matter what. So that is also different than the lab. So my two big questions I answered and wanted to know were, how often do we make saccades and smooth pursuit eye movements during everyday real world activities? And the second one is, how does the type of activity change how we move our eyes? So to answer these questions, I did three main things. The first one was wearing my Tobii eye tracking glasses every day, which collected over 60 hours of data. Then on those data points, I created a lab notebook and I wrote a how-to manual on using the glasses. Next, I put the data into an analysis software, and then I tagged an event based on what was happening in the video. And these are also called activity types, or TOIs, which is time of interest. So those time of interest could included computer work, phone scrolling, walking outside, walking inside, and sitting outside. There are also could be any other ones that you can create, but those are the main ones that I focused on. And then finally, I used the software to detect the eye moves that occurred during that data. And then I just put them all together, pulled out the time of interest, and then counted how many movements were created during that time. So my three main hypotheses. The first one, I said that saccade rates are higher than smooth pursuit rates across all recording and activity types. The second one was saccade rate is highest during computer work because of the continuous eye scanning and reading. And then the last one was pursuit rate is highest during outdoor walking because of the higher frequency of your eyes moving to watch like a car drive by. So more into my methods, because that was my main thing that I did, was the eye tracking part. So the Tobii Pro 3 glasses are the glasses that I used. As you can see, there's little lines, which are the 8 illuminators that reflect eye through your cornea into your pupil. And it the little cameras record that, and it calculates a gaze point as to where you're looking at in the video. And then I don't know if you can see it, but there's a little triangle, which is the front facing camera. Here's my phone that starts and stops the recordings. It also just makes sure everything is okay. And then this is the battery packet that's connected to the glasses, which has an SD card. So everything's saved to an SD card. It's very lightweight and portable. The next thing was calibrating the glasses. I did this every single time before starting the recording. So it's just a little white card that you hold an arm's length away from your face. And then it helps you align to your specific eyes and make sure that it's very accurate based on your eyes. So the next thing I used was my lab notebook. It kept track of anything and everything. So the first thing that I mainly put in was the date, the time it began, the recording name. So it's going to be Aubrey Toby, the day it was, and then the recording of that day. So the first one would be A, etc. And then the folder, which was just basically the name that it was put under, and then the duration in minutes, the gaze sample. So the gaze sample is out of your time, that's how much your eye was actually recorded. So where the circle was recorded. And then your tags were basically just naming what happened during that recording, and then the notes or anything that you observed that happened. So like the glasses slipping. The next thing I did was I took all my data and I put it into the Tobii Lab Pro software, where I put all the recordings together and I backed it up onto a server. From there, I opened up each individual video and I marked the beginning and the end of that video. So an example is computer work begins and then computer work ends, which creates a time of interest. If you can see at the bottom, there's like these white lines. That is the computer work time of interest. So that clipping would just get pulled out and put with the other times of interest. And then the little markers at the top are just like the events that occur. The eye movements, I don't know if you can see it, but they're like little lines that just show if it's recording or not at that time. The next thing are my 2 videos of my computer work and phone scrolling. So these are 2 examples. As you can see, computer work, whenever I read, it goes like across the line. And you can also see my eyes going back and forth to the computer, to the phone. And then for phone scrolling, as you can tell, like the area of interest is very small. So my eyes are going to be trained going up and down following the recordings. The next thing I'm going to show you is walking inside and walking outside. As you can tell, I'm mainly looking in the middle of the video and my eyes are continuously moving like really sporadically quickly. And if you kind of look, you can kind of see where I'm looking and that's where I'm going to go. So you can predict things like that. The final thing, is sitting outside. So if you can see like the red dot goes away, that's what lowers the gaze point accuracy. It probably is because of an error from like the light reflecting and it just drops the signal. Or if you look too far down on the glasses, it also stops recording your pupils because it cannot see it anymore. And then eye movement detection. To get my results, I pulled out the gaze points from the recordings. So out of my 57 total, only 43 recordings had a 90% gaze point and above. And in those 43 recordings, we applied a filter that fills in the gaps of if it drops. And then it just guesstimates what it is. And then if it's bigger than 75 milliseconds, that could be considered a blink. So the filter was not applied. And this happened for each time of interest. It was next put into the robust eye movement detection for natural viewing. This is a Python-based free and open resource software. It takes in horizontal and vertical gaze positions and outputs the time and property of the eye's movement events. So the results from that, it detected over 500,000 total eye movements across 30 hours of the annotated gaze recordings. And from that, I took out 20 seconds of that recording to show you what it looked like. So the Y shows like the gaze position, so basically wherever you're looking at in front of you, where your eye is at. And then the time below is just the pinpoint as to when you did that. If you look at the solid top line is your left and right movement, and then the bottom dotted line is your up and down. The saccades are showed here with the red bars, and then the blue bars are the smooth pursuits and when they occur. And then it calculated the distribution of eye movement event types. So as you can see, fixations occur the most, around 51% of the time. The saccades recorded about 43% of the time, and then smooth pursuits occurred around 5% of the time. So back to my main hypotheses. The first one that I asked was the saccade rates are higher than smooth pursuits, which was supported to be correct. As you can tell from the last one, it said that saccades accounted for 43%, while the pursuits only made up 5% of all events. So in all of these gaze point, only the validity of the computer work, phone scrolling, walking inside and walking outside all seem to be around 90%. So that is a very good recording of your gaze point. But as you could tell, walking outside is a very low gaze point at 74, which again could be because of an error, because I looked down, a blink, stuff like that. The next result that I got was the saccade rate varies by activity. So seemingly there are 1 to 2 saccades per second. And as you can tell, sitting outside is the most. So back to my second hypothesis, my saccade rate is highest during computer work due to frequent reading and screen scanning, which showed to be unsupported. Well, actually computer work was the lowest, but sitting outside had the highest rate. And then my third results that I had were for the smooth pursuits. And it seemed to be that there was a half a pursuit per second, and walking outside had the most. And then back to my last one, the pursuit rate is highest during outdoor walking, seemingly was correct. This is because the highest point was walking outside, and then indoor walking followed that, but everything else was much lower. And then back to overall, my saccade rates are higher than smooth pursuits. Saccade rate is highest during computer work, and the pursuit rate is highest during outdoor walking. The first one seemed to be true, the second was false, and the third one was true. So my conclusions, my key takeaways, we move our eyes all the time, but we mainly make more saccades than pursuits. Different activities change how you make these movements and when you make them. And then interpreting eye tracking data can be difficult because of the amount of errors that can occur. What I would do next. So to test these errors to see if they are an error or if they are true, I would go to various lighting to see if the lighting affects the gaze point accuracy. And then they test how head motions like influence the gaze point to see if it goes up or down. And then record eye data during other activities. So like driving, you would make more smooth pursuits. Eye tracking is a valuable tool to understand eye movements, which can help in clinical settings to monitor brain and vision health. An example is if a patient has epilepsy. It can track if your eyes are creating the deviations of having a seizure, and then that can alert someone to get them help. So thank you. Any questions? And I just want to thank everyone for all their help, and thank you all for organizing this. Yeah.
For a great job. Second, these are cool actually, classes that I have here.
They are very cool.
Yeah.
I'll tell you what, briefly, as Aubrey was presenting, it reminded me of the first day she came to my office and I showed them to her. And I said, these are expensive. I said, are they over $1,000? And they're $20,000. Oh. And I said, yeah. I was like, the first rule is like, don't break them, right? Like, we're gonna, that. So I can get you a pair.
That's big.
No, just.
Okay, check the science before.
It's okay.
Okay, go ahead.
Sorry.
So back to science. So you have this like figure, like the pie graph, where you show you have the fixation pursuit versus saccades. Can this ratio be changed by training? Different people in different conditions.
Yes, it
So
can be
imagine that somebody in a worker in a factory, and they have this kind of pipeline going in front of them, and they have to pick up defective, let's say, product something. You have like a better person, better maybe saccades. Is that?
Depending on what you're recording, if you make more saccades, it will show the rate of saccades being higher. You might not make as many fixations or smooth pursuits, or in a different setting, you might make more smooth pursuits. So it can change depending on what you're looking at and what you're doing in that data. Go ahead.
Do you think this might be biased at all? If it's just from you?
Yeah, it can be, because your eyes, depending on the person, like if you're shy, like you might look at the ground more and create specific motions rather than if you look straight. So it can depend on who you are and what you are looking at. Stuff like that.
Are the glasses really uncomfortable to wear? Do you like constantly think,
so
everyone's gonna see what
there's
I
like these nose pieces, and it like helps like sit on your face better, but like it like one point I didn't even realize they were on me. Like you can't remember. They like kind of like give you a headache, but that's like on like hour 3. So they were pretty good throughout.
So along the same lines as everybody else's question, I notice you're not wearing glasses.
No.
You don't normally wear.
I have reading glasses, but there are like lenses that you can attach if you do wear glasses.
So I guess this might be a complicated question, but how much does the knowledge of, first of all, knowing you do an experiment, that these classes are measuring things, and how much does that influence your saccades and your smooth pursuits? And secondly, you know, is like, you don't normally wear glasses, how much does that affect it? I mean, these are all big questions.
Right.
But
But I
think about.
I do understand what you mean. I think like I am more conscious of my eye movements because I am wearing them.
Yeah.
So I kind of feel like I do probably make more of a certain type of eye movement than the others because I know like I'm wearing them and it's tracking my eyes. And then along with like the wearing glasses, I do sometimes wear glasses, so like it wasn't like really off for me, but like because I was comfortable with wearing them, it was fine. So I think it depends on if you do wear glasses, that can affect if you feel more comfortable like in your eyes, like going with that, if that makes any sense.
I guess this is where you want to come on, somebody that knows nothing about the glasses.
Well, thank
William Zhang.
Okay. So it's my pleasure to introduce Will Zhang, who's been with my lab for like a few weeks. First of all, a little bit about my lab. So I'm very interested in transcription regulation in retinal development. We use mouse as our main model. So my lab, uses many different methods and experiments to test certain hypotheses. And we straddle like different fields like molecular biology, we do like cloning, we also have like histology, we do also cell culture. And I'm really pleased to tell you that actually Will actually mastered all these different techniques in a very, very short period of time. He was like a fantastic lab citizen, been very good in people, asking questions. Everybody was happy with him, And best part of it, actually, he was very productive. He got us some exciting data, and hopefully in the future be able to pursue his data and just get good data. So without further ado, I present you Will Zhang.
All right. Is it possible if I could go, or there's no laser pointer, just use this?
I think this is just remind our old way, I think.
All right, so good morning, everyone. My name is William John, and I'm a rising junior now at Hampton High School. Today, I'm here to talk about my amazing summer project I did in Dr. Aldiri's lab, which is testing the functional validation of the RAX enhancer in the developing retina. I'd like to begin by first talking about the vertebrate retina, which is a structure located at the back of the eye. The vertebrate retina is a highly organized, multi-layered structure that's essential for visual processing and is composed of three principal layers, which include the outer nuclear layer, the inner nuclear layer, and the ganglion cell layer. Right here. These cell layers house 7 distinct cell types, which include the ganglion cells, the amacrine cells, the horizontal cells, the bipolar cells, the Mueller glia, and the photoreceptors, which include the cones and the rods. Each cell type is highly organized and contributes uniquely to the transformation of light into neural signals. Now that we know what the retina is, let us understand how the retina develops within the eye. This picture here does a great job of doing so. As we see, the retina originates from the optic vesicle, which is here, which invaginates into the optic cup here, or a bilayer optic cup. The bilayer optic cup includes the RPE, or the retinal pigment epithelium, and also the inner layer, which includes the neural retina. During the retinal formation, many cells proliferate and differentiate from the retinal progenitor cells, which are basically multipotent stem cells. And these, the RPC layer differentiates into these unique cell types. This image does a great job of showing how these cells come to be. They're early-born cells, which includes the ganglion cell, horizontal cells, the cone photoreceptors, and the amacrine cells. There are also late-born cells, which include the rod photoreceptors, the bipolar cells, and the Mueller glia.
Each of these cell types originate at different time periods during embryonic development and are governed by patterns of gene expression and sequential activation of transcriptional factors. For the purpose of today's discussion, I'd like to focus our attention onto the RAX gene, which is a critical transcription factor required for the maintenance and the specification of the RPC cells. But you may be asking yourself right now, what is RAX? RAX stands for Retina and Anterior Neural Fold Homeobox. It was discovered in 1997 by Dr. Connie Sepko and her team at Harvard University. During this discovery, it was found that RAX plays a crucial role in early eye development of the vertebrate eye. To the right, we see a picture of an in situ hybridization of RAX mRNA in cross-sections of the mouse retina. We see here that RAX is highly expressed during early embryonic stages, more specifically in embryonic stages 9.5, 11.5, and 18.5. We see a lot of staining happening there, especially in the ocular vesicle and also the RPC cells. Later, after birth, we see that the staining slowly declines. This means that RAX expression declines overall. Now, through this image, we're able to tell that it is obvious that the RAX gene plays a large role in the eye development. So it shouldn't be surprising that any mutations and failures of the RAX gene can be devastating, especially to eye development. Here are some examples of the phenotypic consequences that come from RAX gene dysfunction. At the top of the slide, we see an image comparing two mouse models. On the left is a wild type mouse, which possesses the proper RAX, functional RAX gene. And as we can see here, there's this mouse has normal ocular function. On the right, we see a RAX conditional knockout mouse, which is a mouse without the RAX gene. As we can tell here, this mouse is present with a case called anophthalmia, which is the complete absence of eyes. Similar developmental defects are also seen within humans. There are clinical cases of anophthalmia and microphthalmia. There are both diseases that are linked to RAX gene mutations. All right, now that we established the critical role of RAX within the human eye or the vertebrate eye development, it becomes essential to understand how is the RAX gene expression regulated. And with that, we had to turn our attention to enhancers, which are non-coding regulatory elements that modulate gene expression by using a variety of co-activators, transcription factors, transcriptional machinery like RNA polymerase II to basically guide gene expression. This has led me to my hypothesis. Do the RAX enhancers regulate the RAX gene expression during retinal development? In order to test this, we first had to find the proper enhancers to test. So we referred to a previous paper published by Dr. Assam in 2017, which shows a sequencing of the mouse in the E14.5 stage. Through this analysis, we see strong peaks at several RAX enhancers, especially at EN2, EN3, and EN1. For this project, I tested RAX enhancer 3. And in order to test RAX enhancer 3, we used a variety of methods and techniques, which include primer designing, cloning, polymerase chain reaction, or PCR reaction, mouse retinal dissection, and electroporation process. We began this entire process by first cloning the RAX enhancer 3 into the Stagia 3 vector. This image does a great job of showing the overall cloning workflow of this entire process. We initiated this process by identifying the RAX enhancer sequence using the UCSC genome browser. Then we imported the corresponding DNA sequence that we got from there into an application called SnapGene, where we designed the forward primer and the reverse primer here. And after we finished the primer design, we moved on. We got the target DNA sequence then we moved on to amplify the DNA sequence using a PCR reaction. After that, the PCR product was then digested using the restriction enzymes ECOR1 and SAL1. Subsequently, that digested insert was then ligated into plasmid vector Stagia 3 using DNA ligase. And after that, we used that product and we placed it into competent bacteria cells. In our case, we used a DH5 alpha through a transformation process. After that, after the cloning process was all finished, we moved on to electroporation process, which was carried out to introduce the enhancer genes into the mouse retina. To prepare for this, we first prepared a DNA solution comprised of the enhancer plasmid and also the mCherry plasmid. We also, while this was happening, we also dissected a E14.5 mouse retina. After this process was done, we combined both the mouse retina and the DNA enhancer solution into electroporation chamber, where electric voltages were sent to facilitate the uptake of this DNA into the mouse retina. And these are the pictures. These pictures up here illustrates what we want to see from this electroporation process. What we want to see is a really nice dark red fluorescence in the mCherry signaling. This basically tells us the electroporation process was extremely efficient. We also want to see a nice dark green hue in the GFP signal and a nice dark purple color in the AP staining signal. Both the GFP signal and the AP staining signal tell us that the enhancer is driving activity within the retinal cells. These are the results I got from my experiment, or the electroporation process of RAX Enhancer 3. So as we can tell here, there's a nice dark red hue in the mCherry signal. This means that our electroporation process was extremely efficient. We also see a nice dark green color in the GFP signal and a nice dark purple color on the AP staining signal, especially on the peripheries where the RPC cells are located. Both the GFP and the AP staining signals tell us that RAX Enhancer 3 is driving activity in the E14.5 mouse retina. So there are some major conclusions that I could reach through my project. To begin, there was a successful cloning of RAX Enhancer 3 DNA into the Stagia 3 vector. This was proven through a sequencing process. Next, the ex vivo electroporation showed robust and localized reporter activity. And finally, the most important conclusion I was able to reach through my project was that RAX Enhancer 3 is active within the E14.5 retina. Overall, this allows us to further understand the roles of the RPC genes and eye specification, and this could further help us improve our understanding of eye development and evolution. There are many future directions I would like to take with this project. To begin, I would like to create a RAX Enhancer 3 conditional knockout using the CRISPR-Cas9 gene editing system. Now, using this, I would like to see how are downstream transcription factors affected by this conditional knockout. Another direction I would like to take with this project is testing the functional significance of other RAX enhancers, more particularly RAX enhancers 1 and 2. As we saw in the image before. I'd like to end off this presentation by giving a few acknowledgements. I would first like to give my sincere thank you to the members of the Alderi Lab, especially my mentors, Dr. Assam Alderi and Dr. Kamashi Mehta, for helping me with their support and guidance throughout my project. I would also like to thank the UPMC Hillman Academy and Dr. Chen and Kate for giving me this amazing opportunity. And finally, I would like to thank my parents for their consistent support throughout this entire process. Thank you. Any questions? Oh,
Like rats, the rats.
Rats, okay.
The rats.
The without the eyes, okay. This one?
Yeah.
Okay.
So I'm guessing then, what you said on this slide is that RAX basically, makes the eye develop in a sense.
Right.
Is just the simplest way I'm putting it.
Mhm.
And my guess is that when the eyes are not there, the structure of the bones and the muscles that are coming into the genes are differentiating with the absence of the eyes. So has there been a study that has been done on the genes of the organs and the structures around the eye in the absence of RAX and the eye?
Well, I'm not the expert at this, but I assume that it does sort of change it, but the RAX is directly correlated with eye development. I'm
I
not
don't think we know. So.
Yeah.
Because it's outside on.
Yeah, we're not the experts at it. Yeah.
I love your.
But probably almost like certain that function along the eye, because there's no eye, so there's really nice proper structure of like muscles and nerves that actually innervate, surrounding the eye. Probably
Is that thing like on the am I saying that correctly? Is that muscle
No, I would.
I don't know, probably the other thing. That's
Okay.
All right, any other questions?
So for development, it's time is so important,
Right,
right?
definitely.
Thinking about the translation in the future, is there any possibility we can rewind the clock for a patient like with an
Anophthalmia.
or microphthalmia to re-click with some gene manipulation, let the development of the eye happen again? So how far are we if we can give an unlimited resource?
Right, hopefully this will be something that we could do in the future to treat these cases. Yeah, definitely. Any more questions? All right. Thank you, please.
My speaker is Marcus. Mentor is Dr. Kinchington.
So my name's Kip Kitchington, and I've been here for a while. I'd like to introduce Marcus. He's my
There's just a little tweaks and stuff.
a little more.
You send it again or?
He's actually an
Well, I just
undergraduate
uploaded it to the
of
file.
Allegheny
The
College.
Hellman.
Something from his second year to his third year. But when he came to the lab, he had not much experience. I put him on a project that to fill a hole in something that we've done, and we'd done something earlier, and then we brought it out of the freezer, and basically there was a hole. It wasn't what we thought we'd frozen. So I asked Marcus to complete it. He's going to tell you about it.
The one that we had to submit to, but if you can't find it, then we just.
Okay.
did
Oh, I
one
also got
second.
it 2 more weeks longer than you guys. I think it was a lot of fun, actually.
Yeah, that's.
Which one?
That's the-
Marcus, yes.
Because he's an undergraduate.
Yes. So actually Marcus is our first Doris Duke undergrads for the program.
Yeah. Doris Duke Award.
Yeah, so just for everybody, rest of you know, we have undergrad program and a lot of the Hillman alumni keep applying and get continual research opportunities. So you are all welcome back if you want to spend another summer to our site or other human center sites.
I definitely recommend it. I enjoyed it. So my project over the summer, I worked with varicose zoster, which is the virus that causes chickenpox. And my project specifically was creating plasmids of epitope tag ORF62 genes in VCV of both the wild type and vaccine strains. So a little bit about VCV in particular. So chickenpox occurs on the skin of the wild type version. And VCV will actually travel down your nerves in your body and lie dormant in ganglion cells. This can be for many, many years and often decades, where it will then travel back up those nerves and express itself in the skin again, which is known as shingles. So VCV is responsible for chickenpox and shingles, but another thing VCV can do is actually travel up the spine. And sometimes what can happen is it can enter your face region, and from there it can go into your eye, which is our whole rationale of studying VCV. And once it's in the eye, first of all, it's a very painful experience. And it can oftentimes lead to blinding and very bad vision loss. So VCV, there's a vaccine for it, and this vaccine is a live attenuated virus, which just means it's a live version of the virus, but it's very weakened. And in this VCV vaccine strain, there's a bunch of nucleotide changes, like more than 250 compared to the wild type. And the virus gene that these changes affect the most is actually a gene called ORF62. This gene is responsible for encoding for a protein, a very critical protein for VCV called IE62. Basically, what IE62 does is it acts as an activator for all the VCV genes. And without this protein, IE62, VCV cannot grow. So our ultimate goal of my lab is to basically do something called immunoprecipitation, where we'll do this whole procedure laid out here, where we basically use this bead attached to an antibody and pull down the protein. That way we can study the host proteins along with the DNA regions that are associated with the IE62 protein. However, in order to do this, you need antibodies that will attach to this protein. With this protein in particular, we don't really have any good antibodies that will attach to the protein itself. So a workaround is adding epitope tags to it, which is kind of where I come in. And as I mentioned before, this is kind of the variation with the host proteins, and then this is the variation with the DNA regions. This one's a lot more complicated. I didn't have to get into the specifications of it because it's way further down the line. But the basic idea stays the same, where we pull down that IE62 protein and then study the DNA regions that come down with it. So the purpose of my project was basically to contribute to obtaining a library of different epitope tagged ORF62 genes. So those can be used in the processes that I mentioned before. Because again, you need an epitope tag IE62 protein in order to even pull it down and study the things that they want to study. This is a diagram of the library that my lab is trying to gather. The one that I was assigned to creating basically was this HA epitope tag, but there are also 3 others that they're working on gathering. And basically, simplified layout of my project was starting with PCR, basically PCRing the ORF62 gene and adding the epitope to it, which do that for both the V-OCA strain and the P-OCA strain, which is the wild type and vaccine strains. And then from there, clone them into plasmids, where the plasmids will be used as a template to insert that tagged gene into the virus itself. So starting off with the methods, you would start, I was given the ORF62 gene just as is. And like I mentioned before, the first step was PCR. I had to add the sequences for the HA tag, and along with those sequences for restriction enzymes that we use, which were EcoR1 and Bagel2. So PCR, I added the sequences for both those restriction enzymes along with the epitope tags. And once those were added, you basically isolate the product from that and then cut with those restriction enzymes, because these enzymes will only cut if their sequence is there. So that's why we had to add them. And once this enzyme cutting is done, it results in sticky ends, which will then be used to insert in the plasmids. I have a depiction later on that will show that. But the first kind of set of results I got was from the PCRing. And basically with this, as you can see here, these all 4 KB bands represent the fragment that I amplified, and all of them came back positive, which was a good sign. That means the amplification in both times, I did this 2 different times, worked. So it was a good start. Now, after this PCR process, you basically cut out the DNA from that gel because that's what that was. It was a running gel electrophoresis and the screening of it. You would cut out that, elute it, so like melt it down. And preparing the vector we used a vector called PUC48. This vector was our goal of getting the fragment into that vector. And this vector actually has an ampicillin resistant gene. Ampicillin is just like an antibiotic. So bacteria that have this vector will grow on ampicillin plates, which is important for later on in my procedures. But after you prepare the vector, you ligate the fragment with the cut vector, and then through a process called transformation. That's when you put those plasmids, those complete plasmids with ideally your fragment inserted into the vector, into bacteria, clone them, grow them up, grow up colonies, pick out those colonies, grow up cultures from those colonies, then purify that DNA down, cut them again with the restriction enzymes that I mentioned before, EcoR1 and Bagel2, and then basically confirm whether or not your samples have those plasmids with the insert that we wanted. So again, as I mentioned before, this was like the preparation of the vector. So here's the vector we used. We cut it with the enzymes, Bagel2 and EcoR1, which again resulted in the sticky ends here. And you were able, since they're complementary, you're able to combine them through ligation using ligase and result into this, ideally, again. And this is actually what you would be inserting into that bacterium. And as I mentioned before with the plates, I did a bunch of platings. This was just like one example of them. With these plates, we had 3 controls, no DNA control, so didn't have any of our DNA, like any vectors in that. This was basically testing to make sure the ampicillin worked. So ideally, no colony growth on that one. And in this case, that was good. No colonies grew on that one. The next control was a no ligase control. This basically had our vectors, but no ligase. So we should expect to see no colonies on this one. And in this case, we didn't, which was good. However, our final control, the ligase control with the vector only, ideally, since we didn't have our fragment, our DNA in this control, we should see no colonies, because once EcoR1 and Bagel2 cut, they cannot self-ligate with each other. So the vector shouldn't become whole and the bacteria shouldn't be able to grow on the ampicillin because they won't have that vector with the ampicillin resistant gene. However, we had colony growth on these ones basically every time, which suggested that there was something going wrong in the cutting of these vectors. So basically what can happen is when you're doing the cutting of the enzymes with the vectors, only one enzyme will cut the vector. That could be either EcoR1 cut and Bagel2 didn't, or vice versa. There's no real way of telling, but that wasn't a good sign, and this kind of suggested that my vector didn't cut properly, which was supported through running of gels and screening them. Again, I mentioned I did this 2 separate times. The first time, all 24 samples came back positive for no fragment that I was trying to insert. So these all failed. And again, like I mentioned before, it was supporting the idea that the vectors didn't cut properly, so that insert couldn't be inserted into the vector. However, the second time around, again, most of them failed, but I did get one positive sample for that KB band. If you remember from the PCR, it was also a 4 KB band. That's the length of that DNA. So this was a good sign. So at least got one positive sample with that insert, that epitope tagged ORF62 gene inside that plasmid. So coming to the conclusions, like I mentioned before, we had the one positive sample. So that's a step in the right direction. However, we'd prefer, to have also the wild type and more of the Vioca. Basically, the sample that was positive, we sent off for sequencing, and it came back as a Vioca strain or the vaccine strain. Ideally, you would want to have also the wild type so you can compare them, but also additional screening later on, not done by me because I'm at the end of the program, but it should reveal that they'll have, those wild types with those plasmid inserts in them. And also the cloning for the other epitope tags is ongoing, but that's kind of the bulk of the library that we started with anyway. When we first started, we didn't have any of the HA tags, so this is a step in the right direction. But also something that I just wanted to mention, as Kip mentioned at the beginning, I came into this not knowing like any of the procedures I was going to end up doing, or anything about that, and it was just a great learning opportunity. I learned a lot with like PCR, ligations, working with restriction enzymes, and overall, I learned a lot and thoroughly enjoyed the program. With that being said, I'd like to acknowledge the Hillman Academy for giving me this opportunity, as well as the University of Pittsburgh for allowing me to work in a lab over the summer, and everybody that helped organize, Dr. Chen, Kate, orientation in the beginning. I didn't have that the first time I came here. That was very beneficial for me in conducting my research, and yeah. Any questions?
Your results didn't work the first time you looked at your process.
Yeah.
Okay, so what did you do to improve it?
Yeah, so basically I had to go back. That's the reason for doing 2 PCRs. I had to go back and do the PCR again. I kept the same vectors. Basically, we had to isolate the vectors in the beginning so we can combine the fragments from the PCR with the clean vector. So the same vector was used twice, which was kind of like the first suspicious thing after doing it twice and it's still not working very well. That's actually what I'm doing now. Kip had me go back, remake completely clean vector, remake completely clean PCR samples from the beginning. And I basically did the entire thing through again, this time knowing that my vector was clean along with my PCR samples. And the final gel that I showed you of both, like only one sample working on one case, that's the gel I'm currently staining right now as we speak. So I'm hoping to see more positive samples so we can get, you know, another or like a wild type version. But ideally, since it was clean vector and clean PCR samples, that those more would be positive. That's what we're expecting at least. But again, as I showed you with the colonies, this time around, we did also get colonies in the third control, which again suggests that the vector wasn't cut completely. Not entirely sure why that keeps happening, but hopefully this time get more positive samples this time around.
So did you use the same restriction enzymes?
Yes, the entire way through using eGrower1 and Bagel2 the entire time.
Thank you.
Yes.
I just wonder, since you are doing a lot of biology, so which is most challenging part to do?
I'm sorry, what was
Which
it?
is most challenging part?
Oh, oh God, it was all kind of like a bit difficult for me just because I never did any parts of this. I would say there's the transfection part, you had to be like real quick because we use electricity to zap the bacteria and insert our DNA in there. I think that was the more challenging part for me because I had to get used to moving quick and providing the bacteria that just got shocked with media. So we got the ideal result. But yeah, everything else Kip provided me with like protocols and stuff, and they were easy to follow. And anytime I could just ask him, you know, so. But
You
yeah.
have many texts.
Yeah, yeah. I like
I was stuck in the office. I also got a lot of exercise because I'm running backwards and forwards from my office to the lab.
Yeah.
It'll be good for another spot.
That was a T32, I'm afraid.
Yes, like I mean, Kip is a master of the molecular biology. Actually, he runs our molecular biology course. I think there's many things that can prevent civilization. This chain goes like this, right? You prevent that, you can actually, depends on how you purify your hot vector. So one thing, you know, it's very common to have this cell application. So what you like to make sure is that you only collect and purify the ones that have been cut. And one of the ways to do that, you can run it in the gel. So if it's cut, it's usually linearized and it's running a certain speed, and then you can cut the gel and then purify this and do this the ligation steps. So that helps a lot. There's other stuff. You can change the ratio between the insert and the plasmids. It's really standard. I mean,
That's the vector he has right now
Yeah,
on his gel state.
yeah, I mean,
So I'm expecting everything.
probably, yes. I mean, it's a matter of time, you know, if you have more time, probably Kip will run you through all these different opportunities. She's there. So don't get frustrated. That's very normal. Happens all the time, you know.
Yeah.
Yeah, we all face that.
Thank you.
So our next speaker is Jackie Tan. And the mentor is Dr. Funju Chang. Please come on.
I'm
give
good,
a-
I mentor Jacqueline Tan, or we call her Jackie. So sorry, this is my daughter Rachel, since her daycare is not open today, so hopefully I should start her science. And so we are very happy to have Jackie with us, so she's a quick learner. And she not only finished her project, also have a lot of our other members. So I hope she enjoys the science and also enjoy the time in my laboratory, or hopefully she can start her science journey also from here. So, Jackie.
Hi everyone. My name's Jacqueline, or everyone calls me Jackie, unless you're my mom when she's angry. But I'm a rising senior at Mount Lebanon High School here in Pittsburgh, and I was working in the Chang lab this summer. So my project mainly focused on exploring the role of IGF-1 in retinal ganglion cell neural axon outgrowth in a human stem cell derived organoid model. So first going over the entire topic of my project, which are retinal ganglion cells, or RGCs. RGCs are part of the retina, and basically what they do is transmit all the information that's gathered by these purple photoreceptor cells, and then transmits them through their structures called axons through the optic nerve and into the brain to allow you to see. But these RGCs are very easily and commonly damaged by diseases like glaucoma, and this causes irreversible vision loss and blindness because, as I said, RGCs are neurons, and in mammals, neurons are never able to repair or heal themselves on their own. And right now, there are no treatments for blindness caused by RGC loss, so we're working towards RGC replacement therapies where essentially we culture our own RGCs and then implant them into the eye to hopefully get to replace the old RGCs. So, and then in my project, we looked at IGF-1 growth factor, which deals with two main pathways that have to do with differentiation and axon growth. I mainly was concerned about that whole axon growth part, but essentially IGF-1 helps the RGC cells to grow healthy axons and keep the axons from collapsing out. So onto our methods. So I used human stem cell derived retinal organoids, as I previously mentioned. And what you might be asking, what are organoids? So organoids are 3D cultures of cells that are made to resemble a certain tissue. So like in the name organoids, it sounds like organ. So they're you can think of it as kind of like a mini organ. So you can see one here, ours are obviously resembling the retina, and they were harvested at day 56 and day 57. So the first model that I decided to use to test the effects of IGF-1 is basically we have a standard 12-well plate, and we have 2 wells, and in each well we put 1 organoid. And then in the well on the right side, we put in IGF-1, the growth factor, and then on the left, we placed PBS as a control, so we could compare the effects of IGF-1 against kind of normal growth. And then in the second model, we used this multi-chamber chip that has a well in the middle and then 10 wells surrounding it with many channels connecting it. And I have a zoomed in picture so you can see a little bit better. But in this one, this allowed us to kind of see the growth more clearly and compare the growth of the different axons. So we put the organoid in the center, which you can kind of see in this picture, but it's not really clear. And then we put our growth factors in the surrounding well. So similarly to the first model, we put IGF-1 and PBS, but we also tested another chemokine called CCL5, which has been shown to also upregulate axon growth, just as another comparison to see how effective IGF-1 is, since you know, if it's effective, but not as effective as another growth factor, then it's not really worth continuing. So after we implanted the organoids, a beautiful picture of me, I did some imaging at the screening scope using RFP, our red fluorescence protein. And then you can see on the next slide our results. So day one, so you can see the red part is what we're looking for. So as I previously mentioned, we used RFP or red fluorescence protein imaging. So the protein TD tomato, very properly named, indicates where the RGCs are. So you can see here, it's not super clear on the first day, but if we look on day 2 and day 3, you can really see how the axons grew out of the organoid and are indicated by the red light. And then we go on to day 4, day 5, and day 6. Oh, day 6. So you can see really how quickly these axons were able to grow in a relatively short amount of time. So at like day 4, I would say the growth is relatively similar, but at day 6, it's kind of covered up a little bit. But you can see how the IGF-1 axons were able to keep growing and keep staying nice and long, while the PBS ones, they were still growing. However, they kind of collapsed back in on themselves. So this kind of shows how IGF-1 is able to maintain the axon growth. And then on to the next model, which is the multi-channel chip or multi-chamber chip. So again, this is like the center well that I pointed out in the previous method slide. And then you can see day 1 and day 2. Again, you can kind of see how it's mostly growing in like this part. And that's because I didn't really put the organoid in the direct center of the well, even though I really tried, but that's okay. You can see again how quickly the axons are able to grow in even just like 24 hours. And then day 3 and day 4, again, pretty significant growth. Day 5, and finally day 6. So this is our final day. You can see kind of out here these like circle shapes. Those are the outer wells, so you can really kind of be able to visualize that even within like a week, the axons were able to grow a pretty significant amount. And you can see here how our 2 different growth factors, IGF-1 and CCL5, especially looking at these axons, how they were able to grow, I would say significantly more than the PBS axons. However, it's not super clear between IGF-1 and CCL5 which one was more efficient. And so in conclusion, we found that IGF-1 does promote RGC neural axon outgrowth in human stem cell derived retinal organoids. So IGF-1 should be considered a possibility for future treatments using RGC replacement therapies. So in the future, we hope to continue comparing IGF-1 to other growth factors to find the most effective growth factor at contributing to axon outgrowth. And also we should be able to potentially combine the effects of these different growth factors to maybe amplify their effects. And also we wish to develop a method to more accurately quantify axon outgrowth, especially looking at in like the first model with the 2 different organoids. The 2 organoids are, you can kind of say how they were different shaped. So it's not super clear on how to quantify and how to say if the axons were longer. And of course, the final like overall goal of all this work is to develop RGC replacement therapies in order to combat or reverse RGC loss caused blindness by like glaucoma in human patients. And I'd also like to touch on some other things I was able to help with this summer. So I helped Jiu in our lab do some genotyping for his animal part and PCR for his animal experiments. And I also learned how to maintain cultures of stem cells and organoids with Yalin in our lab. And then I did some more imaging for a bunch of different projects in our lab. You can see that, and was also able to meet a lot of great friends this summer. And I also got to see some very vicious mice. And so finally, thank you so much to the Chang Lab for having me this summer, especially Dr. Chang and Yaling for helping me with my project. And of course, Demetrius, to Caroline for being just very nice and welcoming. And of course, thank you to Dr. Chen and Kate for having a very well put together experience for us here at the Vision Institute. And also thank you to Dr. Boone and Stephen at the Hillman Cancer Center Academy for just providing this really, really great experience for us. Thank you.
So I have a question. So, have you tried those axon treatment, growth treatment all at the same time? Do you expect this effect will change when the age of the organoid change? Have you tried different time points?
So when the age of the actual organoid,
Yes.
I would expect that it probably would have an effect, based off of the actual age of the organoid, because if the organoid's it has all of the cells in the retina, so it has RGCs and photoreceptors, so it has to wait for all the cells to develop. So obviously, if we want to test the length of the axons, but not all the RGCs are developed, then see different growth.
So you did your multiple factors. Does that have NGF in it? Nerve growth factor?
No.
Because in the models that we do, we use nerve growth factor to get axons to grow very well through channels. I just wondered if you had an NGF.
No, but in our also too, we have 10% FBS. Yeah, but not NGF.
I want to put NGF.
Yeah, I think NGF is one, but I think we have a lot of candidates for spectrum test. Happen to pick on IGF-1, we are testing the others. It's a combination to do.
All right, Jackie, nice presentation. I have a question about the axon.
Yeah.
So is it controlled? Do you control how it grows, or do you just let it go?
So in this sorry, if you go back, if we look at this the second model that I used, you can't really see it here, but there's different there's small channels or roads for the axon to grow, so we wanted them to grow through the channel. But in the first one, it's just in a dish, so it's kind of allowed to more freely grow in a sense, if that's what you were asking.
Yeah.
Okay.
Lunch is ready, and we're all gonna have lunch here while the mentors, please come by. Let's vote for the best research award and the best presentation awards. I'll bring the lunch here.